morning, everybody. Sorry, I'm a couple minutes late. Hopefully you can hear me okay. Let me share the screen. Thank you. Yeah, I'm here in Brooklyn still, in case you can't tell with my very ornate background. And so, yeah, sometimes I sometimes have some internet issues I need to work out, but oh, we are good to go. Okay, let me pull this up. Okay, cool. Sweet. All right, so hopefully everybody can see the chat box. Oh, thanks, Brittany. Brittany says she likes my shirt. Thank you. It's fun, right? It's colorful. Some people have been emailing me about the phenology assignment. Just want to let you guys know, like, don't overthink it, okay? It's phenology assignment's supposed to be fun. It's supposed to make you guys go outside and look at plants and get excited about plants and like see them change as they wake up for spring. So yeah, read over the description that is posted, the instructions that are posted on Blackboard. One question that I've gotten a couple times in email is about pictures. So let's put it this way. You're going to be observing these plants over time, right? That's the whole thing. That's the whole point of the phenology assignment, is to observe the plants over time and hopefully see some really cool changes, right? Like leaves showing up out of almost nowhere, right? Or if you're lucky, flowers showing up. Like you're going to be looking at this basically sticks for a few weeks. It's just going to be sticks. Nothing's going to be happening. And then all of a sudden, wow, stuff starts happening. Flowers show up. Super exciting. For day one, or the first, or I should say week one observations, the point is I want to make sure that you guys are actually taking the photos, right? That's why I ask you to include a picture of yourself in addition to a picture of the tree's habit, or the shrub's habit, if you happen to be using shrubs. And then a close-up picture of the bark. The reason why I ask you to do the bark is because, as I said before, this time of year, most of the plants don't have any leaves on them. And so it's really hard to identify, even for me, even a plant person, it's really hard to identify what species you're looking at. And so the bark picture is one way that I could actually identify what species you're looking at, because you could actually tell trees apart by their bark, believe it or not. So yeah, first week of observations, picture of your tree's habit, like its form, picture of the tree bark. If somebody else is taking your photos and you can get in that picture of the tree's habit, great. But if you're doing selfies, it may be difficult for you to get like a good picture of the bark or a good picture of the habit if you're doing a selfie. So feel free for the first week to include 3 pictures for each tree, one that includes you. If there's any other confusion about the phenology assignment, feel free to ask me here today or to send me a follow-up email. So several of you, I've asked anybody who scored a C, D, or an F on that first lecture exam, please email me to set up a time to meet so we can go over your exam individually. I've gotten some emails and I have a bunch of meetings scheduled today, but if you haven't emailed me yet to set up a meeting, please do so today. All right, let's jump into it. We're in the seed plants. Yay! What's great about the seed plants is if we know we're getting close to the end of the semester if we're in the seed plants. So that's always a good thing. And today we're going to talk about Cycadophyta, or the Cycads. Okay. Yes, we know what we did last time, right? We learned about the ovule. And then now we're going to jump right into the gymnosperms. The first of those, which we'll talk about, are the Cycads. Okay, this is our roadmap to get oriented, right? Well into the vascular plants by now. And finally into the seed plants. Okay, so here's what some pictures of cycads. This is what they look like. Let me fix this. There we go. And the name of the phylum is Cycadophyta, which is convenient. It's always nice when the phylum sounds exactly like the common name. So the way you pronounce that is on the bottom here, Cycadophyta. Remember, when you see that ending phyta, you know you're always dealing with a phylum. But with the naked eye, they kind of look a little bit like palm trees. I think that the best way to kind of describe them, similar to like a palm tree. But they are not. They are not palm trees, right? Even though the name cycad comes from the Greek cycas, which means palm, they are not palms. Palms evolved millions of years later. Cycads don't make coconuts. So even though visually similar, absolutely not a palm tree, okay? Not a palm tree. Palm trees showed up millions of years later. In the cycads, there's not that many members. There's about 300 species total that remain today. And a lot of them are actually facing extinction, like so under threat of going away forever. And part of the reasons that they are facing that are because they reproduce very, very slowly. That's one reason. Some animals and humans eat the seeds. And so if you eat the seed doesn't, in this case, doesn't turn into another plant. Sometimes they cause habitat destruction. Sometimes the places where they live gets destroyed. That's the habitat destruction. And some species are actually poisonous to animals, and therefore governments sometimes remove them because they poison livestock. So lots and lots of members are facing extinction. When did the cycads show up? Well, a long time ago, a long time ago, right? Like almost 300 million years ago, during the Permian geological period. But they really had an explosion of diversification in the Jurassic period, so about 200 million years ago. And so that means that, yes, cycads are plants the dinosaurs ate. And if you don't think that that's cool, well, sorry, it's cool. It's cool when plants are eaten by dinosaurs, but also just look at these really old drawings, right? So like people have known for a really long time how dinosaurs and cycads coexisted together. I mean, these pictures are like mad old. Look at the one on the left, right? Little dinosaur eating cycad. Like, oh yeah, delicious. Delicious. Loves it. Sorry, I think these photos are really, really funny. But anyway, or not photos, these drawings. But yes, of plants that the dinosaurs ate, which is cool. Super cool. All right. Cycads, where are they? Right? Where are they? Well, they used to be everywhere. They used to be all over the world. But now they're only found in tropical and subtropical regions. So if you've ever been down to Florida or Central or Southern America, you may have seen cycads before. They also occur in South and Eastern Asia, Australia, and the South Pacific. So you might have seen them there, and also Southern Africa. So maybe you saw them there. There's only one species, though, that is native to the USA, which is Zamia floridana, and occurs in Florida, hence the floridana. And I will say, I'll use this moment to like get on my soapbox a little bit. In terms of plants, the words native So there's two categories of plants, native and exotic. And I don't care for that term. I don't care for the terms. Because in terms of plants, it shows a bit of history with those terms. So for example, in North America, if a plant occurred here before the 1700s, it's considered native. And if it occurred here after the 1700s, it's considered non-native or exotic. And that's a really arbitrary time point to pick, right? Like obviously it has to deal with colonization, right, of Europeans to North America. So what does that really even tell us, right? It doesn't tell us really anything about the plants per se. It tells us about what people decided in terms of a time point. And the words native and non-native or exotic mean different things in different countries, and it always deals with the history of that country. Look what I've just showed you, right? Cycads used to occur worldwide. Let's go back. Oh no, that's here. Sorry, I don't have to go back. Cycads used to occur worldwide, right? So like cycads used to be like quote unquote native, lots of places, right? They occurred worldwide. Then climate patterns changed and different things happened, and obviously they don't occur everywhere now. But I'm just putting that out there. And, you know, cycads, for example, have been around for hundreds of millions of years. And so for us in the US to say something is native because it occurred here before the 1700s, that's a little arbitrary. So I personally don't care for the term. Other plant people get mad at me when I talk about this, but I like to say that something occurs naturally or doesn't occur naturally. That's how I tend to refer to things, but you will see the words native show up sometimes in the slides. It's my soapbox moment. And again, before I move on, it's because there's this connotation that like something in native is good and something non-native is bad. And all I'll say about that is that it doesn't deal with nativeness. It deals with invasiveness. So sometimes something can be quote unquote native and be very invasive and therefore bad ecologically speaking. Sometimes something can be non-native or exotic and not be problematic to an ecosystem. So that's why I don't really care for those terms. Yeah, it is controversial, yes. But I think it's something important to mention because words like native and non-native try to put plants into a good and bad category, and that's not really telling us anything about the plants or the system. Yeah, see, and Vivian said, yeah, when I always thought that when people spoke about exotic, it was invasive. And that's not the case, right? Like there's plenty of things that grow in our region or in our country that are thought of as being non-native, and they don't do anything negative to the ecosystem. And then there's other things that are considered native that are totally invasive and cause a lot of problems. So I like to focus on whether something is invasive or not, and not or its benefits to an ecosystem or not. Yes. Okay. Moving on. Sorry. Soapbox moment. Okay. Cycads. Maybe you have one as a plant at your house. They're sold as landscape and house plants. If you do have one at your house and it's and you need advice about it, let me know. They're often sold as being like an easy to care for plant. They're not super easy to care for in general because they like a lot of sun. But they are often sold. If you live in Florida or California, for example, or other hot areas, you could easily grow them as like your yard plants or house landscape plants. See? Okay, well, we are in the vascular plants, right? So we know that the sporophyte generation is dominant. We know that already because we're in the vascular plants. And we know the sporophyte generation has true roots, true stems, and true leaves. For cycads, there's two different types of habits. So last class, we came across the term habitat. Or no, we've come across the term habitat a lot. not just in last class. But then there's this word habit, and this is something, that's what it is. This word you saw, habit. This word you guys saw when I described the phenology assignment, right? One of the pictures I asked you to take of your different trees is their habit. So I'm just going to write something in the chat box real quick because there's two similar looking words here. And don't want you to get confused. Okay, so habit means form. So like a tree's form, whereas habitat is the location that something lives, right? Like some place where something lives. So they look similar. So that's why I'm trying to pull them apart for you guys. But habit means form. In terms of the cycads, there are two types of habits that occur. One is a columnar habit. So you see here, the trunk of the cycad tree is kind of like in the shape of a column, right? Long and skinny. Other members have this kind of what's called a bulbous habit. And it almost looks like if you've ever planted bulbs, but also kind of looks like a turnip or something like that, right? Like short and stout and round. And we think that this habit, columnar habit, is probably the primitive form. So the form that occurred first in evolutionary time. Okay, so there's some terms here that are going to be familiar, hopefully, because we already covered the ferns. Oh, and hopefully you turned in your assignment this morning on the ferns. If you did not, please send me an email today so that I could reopen the assignment for you, so you can get some points. But there's some familiar terms here. I'll change colors to blue. Why not blue? Okay. So the leaf, right? Leaf, aka frond. So one whole leaf. Here's one whole leaf. Oops. Here's one whole leaf, right? AKA frond. AKA frond. Green part of the leaf, which is that part, is the blade or the lamina. Either term is correct. The stem underneath that, right? This part. There's another one here, right? There's another one here. It's called the petiole, just like we've seen before. And if the petiole, when the petiole extends into the green part, right, extends into the blade like we see here. Another one right here. That's called the rachis. These are terms we've seen already. Okay. Because we are in the seed plants, we're already a little bit more evolved than other plants we've seen. So all the leaves going forward are going to be megaphylls. So remember, when we learned about leaves, the evolution of leaves, those first came about when plants became vascular, right? So our first leaves that we saw were microphylls, right? That members of lycopods had, right? Lycopodium and Selaginella had microphylls. But everything else after those, everything else has megaphylls. Megaphylls, one of their main things about them is that they have multiple veins, right? Not a single vein. Here are some different leaves from cycads. And so what is this leaf shape here? What is this leaf shape here? Okay. All right. So a couple things here. So people are writing pinna, people are writing simple, and people are writing pinna compound. So pinna compound is getting closer to what we're looking for here. So first of all of these, yes. Okay. So somebody wrote in the chat box, pinnately compound, correct. So, or pinnate is also a way you could describe these leaves. That's correct too. So one thing you should look for, right? Remember simple versus compound. Let me remove this. Simple versus compound. Remember I told you if you're like playing with a leaf, yay, I'm playing with a leaf, and you're holding the petiole or the stem, if there's a lot of things above that stem, you're dealing with a compound leaf, right? So in all of the cases shown on this slide, but this one is the most easy to see, that's a lot of things up there. That's not one thing. Here's a picture of one thing, right? That's a simple leaf right there, because there's one thing on the top, right? One thing on the top. But this thing here has many things, right? There's a thing here, there's a thing here, right? Lots of stuff. So we know it's compound leaf. Then we have a few choices when we get to the compound leaves. We have palmately compound, which looks like a palm. And if something is palmately compound, just draw it here. All the things are going to come out from the exact same spot, kind of like a hand, right? Like everything, every part of my hand comes out of my wrist, right? Palmately compound. Whereas pinnately compound is our other option, and it looks like a feather, right? And you would agree. This looks like a feather, right? This looks like a feather. This totally looks like a feather, right? Even this one here. And maybe this one doesn't look the most like a feather, but it definitely is pinnately compound. And to be even more correct, I'll write it in the chat box. If you're pinnately compound, you could have different levels of how many divisions you have, right? And as somebody wrote in the chat box, a few people wrote, this has one division, so this has pinna, right? That's one pinna. There's another pinna right there, right? Each of those things is a pinna because we only have one level of division. And so because of that, you could also say this is 1 times pinnately compound, which I just wrote in the chat box, because it only has one level of division. Yep. There's examples of 2 times and more than 2 times pinnately compound in the fern lectures. All right. Yep, here's just so you could see it more clearly. That's that same leaf, but like bigger. Well, a version. It's not the exact same leaf, but the same species. This is just to remind you when you're looking at the slides later. Right, so this is 1 times pinnately compound. It only has pinna, right? Doesn't have subpinna, doesn't have pinnule, doesn't have any of that, just pinna. All right. Oh, cycads, you know, they, like I said, they kind of look like palm trees, so they don't usually have any real branching, but sometimes the really old ones might, and they almost don't even look like branches. So like this thing coming off here, that's actually a branch. This thing coming off here is actually a branch. They don't look maybe the way that you might think a branch looks, right? They tend to be kind of round. Oh yeah, so cycads, another cool thing about cycads is that they are evergreen. So we saw this term also, I'll change colors, spice it up. Saw this term also last time when we talked about the phenology assignment, right? Deciduous and versus evergreen. So I'll just write it here, deciduous. The leaves fall off each year. Evergreen, leaves stay on for many, many years. Yes, so cycads are evergreen, and they don't shed their leaves every year. Most of that is also because they live in these kind of tropical environments, so they don't really ever experience freezing temperatures. However, eventually leaves get old and have to die, and when that happens, it's usually the lower leaves will die over time as the younger leaves grow from the top, And so a lot of times it kind of forms these like interesting little looking skirts in nature. So again, if you have one of these as a house plant and the lower leaves of your cycad aren't doing so great, that's normal because they make new leaves from the top. And as they make new leaves from the top, the lower leaves will die. So if you have this as a houseplant, you can feel free to remove those dead lower leaves for aesthetic purposes if you want. Eventually, they will fall off on their own. When they do fall off on their own, they leave these really cool, almost looks like dinosaur skin on the trunk. So here's a close-up. Maybe I'll change colors. Here's a close-up of a trunk. And each of these things that you see was originally where a leaf occurred. So all of these things used to be leaves, at some point. And now it's like this armor, almost like scaly armor, which is probably useful. If dinosaurs are trying to eat you, it's probably good to have some thick, scaly trunks. Okay, the roots of cycads are a taproot system. We haven't really talked about root systems yet, but just know what a taproot system is. It means that the root looks kind of like a carrot. So really, really long and deep, really long and deep. There's other types of root systems, but we'll talk about those later. Oh, but no, here we go. Big scary word. Big scary word. So the roots are a taproot system, and sometimes the roots are what are called apogeotropic. Apogeotropic. And let's see if I tell you guys that let me see if do I tell you, like, do I break it down for you? Yes, of course I do. Of course I do. So sometimes the roots are apogeotropic. You also might see, depending on what book you're using or whatever, you might see this word, coralloid. Coralloid. I like apogeotropic better because apogeotropic actually tells you what they're doing. And we'll see right now. But so apogeotropic or coralloid means that the roots grow out of the soil. The roots grow out of the soil. Here is a picture. I'll change colors to blue. Here, see this kind of almost looks like a mushroom looking thing? Those are apogeotropic roots. Apogeotropic roots that will grow out of the soil. So why do I like the word apogeotropic? Because it tells you what's happening in the word. So apogeotropic means to turn away from the earth, right? Or grow out of the soil, away from the earth. Apo comes from the Greek apo, which means away from. Geo comes from the Greek word that means earth. And tropism refers to tropos, which means like the troops, I think. But anyways, it means to turn or to move. So it's these are roots that move away from the earth, or in other words, grow out of the soil. So I like that word. Plus fun to say. Yes, so cycad roots, taproot system, and sometimes the roots grow out of the soil, which we call apogeotropic. Here's a picture of them. Here's a picture of them, right? See, in this picture, those apogeotropic roots almost look like peanuts or something. Kind of looks like peanuts. It just doesn't look like anything you would normally see on a day-to-day basis, right? Unless you're living around a lot of cycads. And if you didn't know better and you saw this coming out of the bottom of your cycad, you might think something is wrong, right? Kind of doesn't look like, what is that? But those are actually roots coming out of the soil. And the reason why cycads have that is because you guessed it, they have a symbiotic relationship. And so those apogeotropic roots, inside of them live cyanobacteria, which help to provide nitrogen for the plant. Because remember, nitrogen exists in the environment in a lot of different forms. The most dominant form is atmospheric nitrogen, so nitrogen in the air. And nitrogen in the air is plants can't just like grab it out of the air. They need that nitrogen to be in a different form, right? Or they need that nitrogen to be fixed. And so what the cyanobacteria do is they take that atmospheric nitrogen out of the air and then turn it into a type, or fix it into a type of nitrogen that the plants can use. So cyanobacteria provides nitrogen for the plants, whereas the plants, in particular the plant roots, give the cyanobacteria a nice home. Nice home. So on the left here, you have a picture of an apogeotropic root, kind of like close up. And in the middle, you see a cross section through apogeotropic root. And you may even see here, see this, I'm drawing it in blue. See this like ring inside? That's that cyanobacteria living right inside. Look, here's another one right here. Lives right inside. And if we were going to look at that under the microscope, see here, cross section of an apogeotropic, aka coralloid root, showing the cyanobacterial zone, CZ, right? So here you go, CZ is right here. And that's where the cyanobacteria live, inside of that plant root. So symbiotic relationship. Oh boy, how much growth do we talk about? My favorite topic. Oh yeah, oh gosh. Oh gosh, yeah, we will not get through everything today, that's for sure, I don't think. But that's okay. But whatever we don't get through today, we can pick it up after spring break. Okay. We have to talk about meristems. We have to talk about meristems a little bit because we're going to learn about a new type of growth today. So far in this class, whenever we have talked about growth, we're always usually talking about this thing called so far we're always talking about this thing called an apical meristem, right? What's the way to? So here's how I'll get you oriented. A meristem is like a region of cells that can divide and turn into other types of cells. So in the past, I've said, you can think of a meristem as like stem cells, because hence why they have the word stem, meristem, right in there, because it's like they are meristem cells, stem cells. So cells in the meristem, there's different types of meristems, but cells in the meristems can grow and turn into other tissues. And then those tissues can grow and divide and turn into even other tissues. This is like talking about body development, right? So apical meristem means apex, like at the tip. So in the past, when we've looked at, like, let's say this is a plant. Look, here's little leaves, right? Has leaves, fine. Whatever. Maybe down here is the roots, right? Here's some roots. When we say apical meristem, we mean that it's at the tips. So like, for example, there would be an apical meristem here. And that apical meristem would give rise to tissues that would ultimately produce either more stem tissue or maybe even a leaf, right? Because remember, they're like stem cells. But apical meristem could also be at the tips of the roots too, right? So up here, this Apical meristem is going to give rise to the shoot system, or stems and leaves. And then the apical meristem down here would give rise to roots, right? But the point is apical. So I'm trying to show you that apical means at the tips, at the tips. So we're going to learn a few terms today about what tissues different meristems produce. So the apical meristem gives rise to primary meristems. That's what the apical meristem makes. And the primary meristems are going to make primary tissues. And we'll see this coming up here. We will see this slide a bunch of times going forward in the course, but I'm just getting you familiar with the terms today. Primary meristems, there are three primary. So apical meristem, which is here on the left, the cells there will divide and become these primary meristems. And you have protoderm, ground meristem, and procambium. Well, a few of these things might look a little familiar with our eyes. For example, this derm, we've seen like epidermis, and ground meristem may remind us of like ground tissues, right? This last one though, procambium, is a little different, like newer for us. So these three here are those primary meristems, and each of those primary meristems is going to produce different tissues, primary tissues in the plant. So the protoderm is going to make epidermis. The ground meristem is going to make your ground tissues that we've talked about before, like cortex, for example, or pith. And then procambium is going to give rise to your primary xylem and primary phloem tissues. This is a slide we saw way back in lecture 4 about these tissue systems in sporophyte plants, right? This is just a recall or a reminder slide to show you those different tissue systems. Dermal tissue system, which occurs in all parts of the plant and is the epidermis, which now we know is made by the protoderm, meristem. Vascular tissue system, which is xylem and phloem. And if you're talking about primary xylem and phloem, we just learned that the primary meristem, procambium, makes those. And then ground tissue, which if you're in a leaf is a mesophyll. If you're in the stem and the root, the tissue is a cortex, which is composed of different cell types. And now we just learned that that's made by the ground meristem, right? That's why I'm connecting those together. Okay, this is why we're talking about all of this. Because I keep saying primary, right? Plants have two different types of growth. And because all the plants we've dealt with so far have been so small, we haven't really had to deal with this, these two different types of growth. So plants have two types of growth. They have primary growth and they have secondary growth. Both types of growth serve different purposes. Primary growth, if you look here on the left, is the purpose of primary growth is to increase length. And that's because primary growth is also due to those primary meristems that are made by the apical meristem. So look at this little tiny tree on the left, right? This little tiny tree needs to get taller. It's growing. And so in order to increase in length, it has apical meristem at the tip and apical meristem at the roots. You can't see the roots here, but imagine there's roots down there. And those apical meristems make those primary meristems that I just described. and allow the plant to grow in length, right? And that's what we just, primary growth is, oops. Oh, geez, hold on. Primary growth is what is, occurs, like I said, at those apical meristems that are at the tips, and those apical meristems that are at the tips produce these primary meristems. Look, primary growth, primary meristems, primary tissue. So that's one type of growth, and that occurs to lengthen the plant. But then there is also secondary growth, and secondary growth is to increase the width of the plant, to make it thick, to make it more thick. Because let's put it this way, if you're going to be a giant tree, right? Like you're going to be 100-foot tree or something, you can't just only increase in length, because you'll get knocked down. You'll fall over. You have to also increase in width in order to get to be a 100-foot tall tree. And the meristems involved in secondary growth are not the same meristems that are involved in primary growth, different ones. So some meristems are involved in getting the plant longer. Other meristems that we haven't talked about are involved in getting the plant wider. Okay. This is important. Make a note. This is important. All plants have primary growth. Every plant, every single plant. Mosses have primary growth. Hornworts and liverworts have primary growth. Everything has primary growth. Now, in the non-vascular plants, their primary growth is a little different because they don't create these complex tissue systems like vasculature, but they still have primary growth. Don't worry about that. The point is all plants have primary growth, but only some plants have secondary growth, which is why we haven't talked about it until today, because not all plants have it. So didn't need to talk about it until today. Let's see how much I want to get into this. Okay. I think what'll happen, yeah, this is I forgot how long this one is. I think we'll probably get through the secondary growth part, and then maybe we'll see how we do with time. We'll see how we do with time. I'm keeping an eye on the time, and we still got some time. Okay, so, right, two types of growth, primary versus secondary. Primary is to get longer, secondary is to get wider. All plants have primary growth. Only some plants have secondary growth. And as we saw on the previous, right? This primary growth is by the apical meristem, meristems at the tips. Secondary growth is by lateral meristems or meristems on the sides. And there are two types of lateral meristem. There is the vascular cambium and the Cork cambium. Vascular cambium, and again, this is a lot of new terms today, but we're going to come across these terms many, many times. Many, many times as we go forward. So don't get overwhelmed. Vascular cambium makes secondary xylem and secondary phloem, which we will talk about. Cork cambium makes cork, also known as phellem, and phelloderm, which we will talk about. This is so this slide I think shows up in a few different lectures, but this bottom part here is a nice summary slide of like the different players. So you might want to make a note of that. because it shows you both primary and secondary growth on a single slide, which is nice. So the stuff on the left, we already came across, right? We already came across this stuff, all the primary growth stuff and all the primary tissue stuff. So all that stuff is the primary. What this is trying to show you is that those lateral meristems that are what make secondary growth are actually produced by the primary tissues. So for example, the cork cambium is produced by the cortex eventually. And the vascular cambium is produced by combinations of the ground and vascular systems. You don't have to know these words. Just know that they're produced, the vascular cambium is produced by some ground tissues and also some vascular tissues, a little combo. So now we have these lateral meristems, cork cambium, and vascular cambium. And as I said on the previous slide, the vascular cambium is going to make your secondary xylem, secondary phloem, and your cork cambium is going to make your cork, aka phellem, and your phelloderm, which we'll talk about. Don't worry about periderm right now. Okay. Oh, or I guess do. Your lab, you'll see in the lab this word periderm. And I think one of the questions in the lab is even like, what is the periderm? Periderm is just a collective term that refers to all three of these things. So if you see that in the lab, that's what that means. Okay. Let's walk through this a little bit. Again, this is my favorite, most exciting part of this class. Let's walk through this a little bit. So I just told you on the previous slides that there's two types of growth, primary growth and secondary growth. Primary growth is via the apical meristem and then the primary meristems that lead to primary tissues. Secondary growth is via those lateral meristems, which were produced by primary tissues. So I'm going to show you what that looks like in pictures. So here in this top drawing, or I guess you can only see this one right now, but in this drawing, you see these are those three primary meristem tissues that were on the first slide about this, right? So apical meristem gives rise to three primary meristems, and those are the ground meristem, the procambium, and the protoderm, right? The protoderm is going to ultimately make epidermis. The procambium is going to make your primary xylem and phloem, and your ground meristem is going to make your cortex and other ground tissues. As this is being developed, look, here you go, procambium just made. So remember, procambium is a meristem that makes primary xylem and primary phloem. Look, there it is. Protoderm is a meristem that makes epidermis. Look, there it is, right? Here's your epidermis now. And then ground meristem is a meristem that makes your ground tissues, right? So like pith and cortex, right? Okay, so now they're there. They've grown. Eventually, don't worry about those words, but eventually parts of the ground tissue system and parts of the vascular tissue system are going to make the lateral meristems. And I'll switch colors to make the lateral meristems, which are called the vascular cambium. Oh, this one only shows vascular cambium so far, which vascular cambium is going to make your secondary xylem and secondary phloem. So look, here it is. It already made the secondary xylem. Here's the secondary phloem. And so that's the vascular cambium meristem. There's another meristem, which is the cork cambium. And I guess they didn't put it on this slide, which is annoying. But the cork cambium is going to make the other secondary tissues. But I don't want you to get bogged down right? I don't want you to get bogged down on the terms today. What I'm trying to describe to you is this. All plants have primary growth. Some plants have secondary growth. And if you're a plant that produces secondary growth, you started off by producing primary growth. So you go from primary growth, tissues and meristems, and then at some point in parts of your plant body, you transition to this kind of secondary growth tissues. And that's what I'm trying to show you with this. Go from primary growth to secondary growth, and it's a transition. And that's what I'm trying to show you in these slides as well. This here in A only shows you primary meristems. This here in B shows you some primary meristems and also some primary tissues. This here in C, don't worry about those terms, but is that transition point where now this plant, in this part of this plant, we're transitioning from primary to secondary. Here we go. And this is going to allow the plant to get wider, So here on this D, we have a combination of primary and secondary. There's a combination, meaning, look, we still have primary phloem and we still have primary xylem. But now we have vascular cambium. So we have secondary phloem and secondary xylem. Actually, here, I'll make it more clear this way. With yellow, I'm going to highlight anything that's primary. Pith, primary. Primary phloem, primary. Cortex, primary. Primary xylem, primary. And epidermis, primary. With blue, I'll highlight secondary. Secondary phloem, secondary xylem, both of which were made by the vascular cambium, right? So on D, we see a combination of primary and secondary things. But by the time we get to E, we're losing a lot of the primary tissues. We're running out. We mostly have only secondary things, right? So there's a little bit of cortex left, which is primary. And there's a little bit of epidermis left, but it's ruptured now and it's gonna go away. And instead, we mostly see here the secondary xylem is the red stuff. So like all this red stuff is the secondary xylem. Secondary phloem is the blue stuff, which we also see down here, which is secondary, right? It says it in the name, and then the phelloderm. So the point is, by the time you get to E, you're mostly dealing with secondary stuff. The reason I'm bringing this up is because eventually, in plants that do secondary growth, eventually in certain parts of those plants, there won't be any primary tissues remaining, right? And then we're talking about the stem, but there won't be any primary tissues left anymore. Eventually there will be only secondary tissue, right? For example, if you're 100-foot tall, very, very big, giant tree, if you were to look at the cells that are in that big, giant tree's trunk, there would only be secondary tissues. Okay, look, got a question. Do I have to understand where secondary growth occurs? Because I don't get it. So you ultimately will have to understand it, but you don't need to today. Sorry guys, hold on. I have like a, so secondary growth and primary growth both occur in the stems. But for today, I'm just trying to get you comfortable with the idea that there are different types of growth, primary and secondary growth, and that all plants do primary growth, only some plants do secondary growth. There are cells and tissues involved in both types of growth. And if you are doing secondary growth, eventually you won't have any primary growth in those structures. But in terms of understanding it completely today, don't worry about that because we're going to talk about this more. But like these pictures, for example, are all cross sections of a stem, And the reason why they're cross sections of a stem, right? So remember this slide here. Let's use the same colors. This stem here wants to get long. This stem here and roots wants to get longer, right? And so that needs primary growth, and there will be apical meristems that allow that to occur. But this tree on the right needs to get thicker and wider. So like, see how thick this trunk is compared to this trunk? The only way that something in the plant world can become wider or thicker are with the lateral meristems or secondary growth. So primary growth is occurring at the tips to get longer. Secondary growth is occurring in lateral meristems that are inside of the stem. But it's not necessary that you understand this completely today. We'll talk about this a lot, or we'll talk about it more in subsequent classes. So back to the secondary growth thing. So cycads, which is what we were originally talking about, they do have secondary growth, but it's very limited. What does that mean? So, okay, let me just go back real quick. Actually, this is not great. Okay. Just know today, cycads do produce secondary growth, but that it's limited. It's limited. And what that means is, okay, so when you think of like secondary growth, think of a trunk that's a trunk of a tree that's trying to get wider, right? And when you think of the trunk of that tree, what do you think about it, right? Like it's hard, it's woody, right? It's like made of wood, right? So cycads, if you were to chop down a cycad tree, the trunk is not like super woody. It's kind of like not so dense. Like you wouldn't want to make your house a big house like out of like cycad wood because it's kind of not super strong. And that's because they do have secondary growth, but they don't produce that many secondary tissues. So they have limited secondary xylem, limited secondary phloem, and limited cork and phelloderm. So they do it, but they don't do it as great, or they don't have it as much as we will see in the other phyla that we'll cover. Okay, let's just see where we're at with this. Oh boy. Okay. I think we'll get through this and then we'll break. All right, so you remember this really fun slide, right? The stele. Oy, the stele. Just can't get away from it. So remember that the stele, again, we're talking about the stems, we're talking about growth, we're talking about tissues that are inside of stems and tissues that are responsible for different types of growth. All right, so we know that the stele is a way to describe how xylem and phloem are organized, right? We've covered this already here, xylem and phloem, and there's 4 types of organization: protostele, siphonostele with no leaf gaps, siphonostele with leaf gaps, and eustele. We've talked about these before, right? When you're in a cycad, if primary growth is happening, you have kind of like this eustele-like siphonostele, which we covered in the horsetails, in that the vascular bundles are in a ring, but there's also an area of pith. So the eustele part is, vascular bundles are in a ring, right? So, right, each of these little rods will be in a ring like this, but there'll still be an empty area in the middle of pith. So similar to what we saw in the horsetails. It'll show you here. So just so you see, just so you know what you're looking at here, this is a cross section through a younger cycad stem. Younger cycad stem. And see here, let me change. Vascular bundles are, here's one, here's one, here's one, etc. Here's one, blah, 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 all the way around, right? So each of those things I just colored on is like a rod of vascular tissue. So it would have xylem and phloem inside, but these rods are also arranged in a ring. So that's why we say it's like a eustele-like siphonostele, right? Because there's still a pith region in the center. Right, but so here, younger stems show tissues associated with primary growth, right? Primary growth, tissues associated with primary growth. Well, yeah, we see pith, we see cortex, right? We see vascular bundles that have the primary xylem and primary phloem. So those are things of primary growth. Don't worry, we're going to talk about this a lot going forward, so don't feel overwhelmed today. What I want you to see from this slide, though, is remember how I showed you that the leaves leave like this kind of armor? So the leaves would have come out of these stems, right? And so just so you know, like these things here are just the leftovers of the leaf vascular tissue. So like this is where a leaf would have been attached at some point. This is similar to like the leaf trace, leaf gap, right? See, like girdling leaf trace. I'm just telling you this because if you see this under the microscope slide, that's what those squigglies are. Okay. Oh boy. All right. So here's younger cycad stem where we see tissues with primary growth, tissues that indicate primary growth, right? Like pith, cortex. But when you look at an older cycad stem, you'll see the structures that are involved, the structures that are parts of secondary growth. For example, you'll start to see these secondary xylem rings. See, here's one, here's another one. No more primary xylem and primary phloem. And the phelloderm, which whatever, we'll talk about another time. The point is, again, I don't want you guys to get bogged down on the terms. What I want you to take away from today is that there are two types of growth: primary growth, secondary growth, both of which have different meristems, both of which produce different tissue systems. Some plants, all plants produce primary growth, some plants produce secondary growth. And depending on the age of the plant that you're looking at that produces secondary growth, you're going to see different types of structures. That's what I'm trying to get across. Where is the oldest part of the tree? Good question. So the oldest part of any tree, there will be lots of parts on a single tree that might be equally old, but I think it's a safe bet that if you're standing next to like 100-foot tall tree, that the oldest part is basically the trunk, right? Like the base of the trunk. A lot of times, if you want to find out where is the oldest part of a plant, for plants that have secondary growth, you would go to the widest part of the plant, widest part of the plant. Okay. I think, yes, I think we're going to stop there for today, and we will pick up We'll pick up with cycad reproduction. Yeah, we'll do cycad reproduction after spring break. Let me stop this because I don't want to rush through it. And there is a lot of stuff in it. Let me pull up the syllabus though. I just want to see. Pull up the syllabus real quick. All right. All right, so because we're not going to finish Cycads today, originally you guys had a assignment due on Cycads after the break. We're going to push that back probably to the Thursday. That so I'll send an announcement about that at a maybe later today. But the point is that like we didn't finish Cycads today, so we're not going to have the assignment on Cycads the first day after the break. Okay. Does anyone have, oh, I got a question here. Will you hand grade assignment 4? Yes, so I manually grade all the assignments. Assignment 4 was due today. So I will probably be able to grade it by the end of the week. Yep, thank you. Okay, if anyone else has any questions, I'm happy to answer them here, or you can send me an email. I have meetings with several of you today to go over your first exam. If you did get a C, D, or an F on the first exam, please send me an email so we can set up a time to meet individually to go over your exam. And unless other people have any questions, you're free to go. Thank you so much. Have a great spring break. Have a good lab tomorrow. And I will see you guys after the break. So have a good one. Thank you guys. Get some rest during spring break, okay? Don't go nuts. Bye, guys.